Bismillahirrahmanirrahim. Microbial degradation of different chemical compounds require different metabolic processes that operate inside the microbial cells. Those metabolic processes require electron donors and acceptors to operate. So let's talk about different examples and different exceptions in this regard. Now, organic compounds normally act as electron donors in energy metabolism, but this is not always the case. For example, in some exceptions, for example, in fermentation, one part of organic compound is oxidized while the other part is reduced. That's the example. For example, over here you can see here that glucose is being converted to ethanol and carbon dioxide. 4 carbon atoms in the glucose are partially reduced to ethanol, while 2 of them are being oxidized to carbon dioxide. So you can see here सिंगल मॉलिक्यूल है, उसका कुछ पार्ट जो है रिड्यूस हो रहा है, कुछ ऑक्सीडाइज हो रहा है. So exceptions are there as well. The part of the glucose that becomes ethanol is basically electron acceptor part, while the other part which is becoming carbon dioxide is basically electron donor part. So you can see here the electron donor and acceptor parts are still there, but not as a completely different molecules. However, the process still operates. Electron donors and electron acceptors should be there. So that energy can be generated. Now there are so many different halogenated organic compounds which are mostly toxic, and those can act as electron acceptors in so many different microbial processes. Example is of reductive dehalogenation of chlorobenzoate. Let's talk about this one. So as you can see in this diagram, we have this chlorobenzoate, and the reaction is performed by a microbial species DCB1. It has been reported in literature, and the hydrogen here is basically electron donor, and it donates this electron, and this chlorobenzoate is reduced to benzoate, and the chloride group is removed due to action of this microbial species, and this chlorobenzoate which has been generated is acted upon by another microbial species named as BT503, and it then converts this benzoate to hydrogen, acetate and carbon dioxide. The hydrogen can be again used to bring about reduction of more chlorobenzoate by the DCB strain. The hydrogen can also be used to reduce carbon dioxide to methane by another microbial strain PM1. So you can see here 3 different strains are working together to bring about a change, and you can see here overall chlorobenzoate is basically being converted to methane. So it depends on the factors and the conditions that involve this process. If the conditions are favorable and all the nutrients and factors are available, all the physiological conditions are met, then the conversion to methane can be very efficient by these microbes. And you can see here different molecules in different reactions serve as electron donors and acceptors. Now the strain DCB1, it is obtaining energy from hydrogen, which is acting as electron donor in this particular example, and chlorobenzoate is acting as electron acceptor. This example is basically can be called as an example of dehalo respiration. Basically respiration हो रही है, लेकिन इसमें ऑक्सीजन इन्वॉल्व नहीं है as such, और dehalogenation भी हो रही है, so it can be called as dehalo respiration. There are so many other chlorinated organic compounds as well which are known to serve as electron acceptors, so those can also be biodegraded in this fashion. So this was about how different compounds can act as electron donors and acceptors in different microbial processes to bring about degradation of different chemical compounds. We will talk more about these in our next lesson.